Okay, good afternoon. Thank you for coming back. So the purpose of this second lecture is basically to apply a little bit more the basic I showed this morning and actually do what I call version 2 of PRM. It means moving one step further. We have all the benefits of PRM in terms of sensitivity, selectivity. The question is, can we go beyond just regular quantification on the classical way, let's say 1,000 or 2,000 of peptides, and try to use it more for larger screens? So we're not intending to full proteome cover, thousands or tens of thousands of proteins, but I guess there is a niche if we could do something between, let's say, a few hundred and thousand peptides. It could cover quite a bit of needs in terms of biology, following pathways and part of the biological questions, or in the more clinical applications, the biomarkers, right? You always have this first stage, you have tons of so-called biomarkers or biomarker candidates, and eventually only a very small numbers ends up to really be differential and statistically significant. You can move forward in a classical quantitative assay, but you have this early on screening phase where you have to explore and test for a few hundreds or several hundreds. So if we could cover this kind of niche, I think there is still a big demand in terms of applications where it's so far been covered with SRM or other approaches as DIA, or even try to do it in DDA with all the caveats and limitations there. So our question was, can we try to scale PRM experiment and try to address that questions? And looks like, I'm sorry, system doesn't want to talk to me, so. So I think the first part we mostly covered this morning, so I apologize if a few slides will be redundant, but I have to repeat or reassess a few things. And that's really what I want to do is move to PRM version 2, and it has basically two components. The first one is we'll do more on-the-flight, real-time assessment of the data and use that quality control, if you will, or this to make decision and do the next experiment. And in order to measure more, we also in somehow reverse the way of doing an experiment. The classical way, you would look for a peptide, you would detect a peptide, and if it's there, you try to quantify using the internal standard. And sometimes the peptide is not there, then you don't know, is it limit of detection, it wasn't detected, or so on and so forth. So the idea is, if we add the internal standard, we have the internal standard, we add it to the sample, we know it is there, it should be there, and we should be able to detect it. And we work on that point, that first to measure the internal standard and then quantify the actual peptide of interest. I'm really annoyed because of, sorry for that. No, system doesn't want to talk to me, so. Actually, can I move it on the other side? So, I think that's better this way. Sorry for that. So, quantitative experiment, quantitative assays, we discussed it earlier today, right? So it's a couple of issues. It is a challenge, but eventually, if we now really want to quantify even a large number of peptides across a large number of samples, the problems, or one of the problems, is the missing value. So we want to make sure we systematically measure, and if there is no signal, it's not because we missed a point, actually measured and there was no signal above the threshold, right? So this is addressing the problem of the missing value. Complexity of the samples, we discussed it earlier, there's a lot of interferences. I'll also come back to that point later on, because it's not only a mass spec issue, instrument issue is actually equally well a sample preparation issue. Wide dynamic range, we know that. It is pretty trivial to measure relatively medium abundance, but when it really goes in low abundance components, we have to struggle and deal with the backgrounds. So sample prep, I'll come back to that in a minute. In terms of instrumentation and experiment, right, we want large dynamic range, low nanomole or even subatomoles range, and the chemical background is our basically the things we have to find against most of the time. The rest we have already seen earlier today. So sample preparation, and this is just one example. I'm not saying this is the only way, but that's some recent work we showed. There's different methods. You can look at subproteomes, and sometimes there is quite complex chemistry associated to it, but here we did a fairly simple experiment. We do know that IMAC couple to couple has a great affinity for histidine-containing peptide. And we also know that histidine is one of the so-called rare amino acid, which is about 1/5 or 1/6 of the peptide. So we can reduce the complexity of the sample by a factor 3, 4, or 5, depending on the species, by not taking all the peptides, but taking the histidine-containing peptide. Right. So there's also a lot of implication and consequence in terms of proteotypicity and half of peptide per proteins. So as we said before, there is nothing free in these things. There's always a trade-off. But in this way, we can dramatically reduce the complexity of the sample. And it is shown here with this one example. This is the one peptide. It is just a digest. It was from a plasma sample without further purification. You measure that, and either SRM or PRM, you see that you get a very crowded space. And if you try to quantify there, you may see your peptide, but you may have interferences as well. If you do the simple experiment, just going through one column and all the peptide non-histidine containing go and the flow through, which is retained is cysteine containing peptide, you look at that fraction here. Now you already have much cleaner, less complex samples, and our way to measure peak purity is actually look at the fragmentation pattern. So in blue, and you see several of those later on, in blue is the reference spectra or the subspectra, the key ions here. So this is the reference spectra from the pure peptide. And if you look in yellow, you see that the spectra derived here is large discrepancies. So clearly you have interferences. In green, it usually is fairly good match with the reference spectra, means you have little interferences, right? And this is an example, but to us, as we do PRM experiment, detecting the right peptide and checking the purity of this peptide, the good proxy of that is looking at the match from the expected spectra measured under the same condition compared to the experimental spectra. And usually deviation from the reference pattern is a good proxy or good indicator for presence of other species or other component in your sample. The second point we have also tried to address is addressing all the sample preparation, and Christina had a nice talk this morning introducing all the different steps. One thing we have is use two different peptides, the same peptide, same sequence, but two different labels. So one amino acid be the lysine or arginine at the N terminus plus a second amino acid. And basically you have two internal standards you can add at different time in your sample, so early on and later on. And assuming you put it as a concentration of the expected peptide, you would expect a pattern like that. If you have now losses on one of the stages, any deviation or deviation for this ratio is a good indicator of the recovery and the losses in your sample preparation process. So it's just two examples of things you can do or you potentially should also implement if you consider quantification. It's not only about just having the instrument with the best settings to get sensitivity and selectivity, because you could be very precise, a CV of 10% of what you measure, but if you lost 90% of your sample in the front end of the process, what is a meaningful or a very good CV, right? So having a good representation of your initial material or protein mixture is actually an important factor. So having QCs along the road and having some good sample preparation where you assess recovery is a very important factor. So we have seen that this morning. SRM, you collect those, you collect your ion or your different transition, and you have a first proxy to check here. You could look at the ion ratio, as you don't have necessarily full MSMS spectra, but this idea of looking at that is what is going to be key in all PRM experiment. And SRM, as we discussed this morning, has two limitations. First of all, we have low resolution mass analyzer, so we may measure convolution of two or several signals in many cases. And the second point, which is, we haven't discussed it, but it is true in SRM, and the same thing applies for PRM. If we want to be quantitative, you want to measure and you make sure that the way you sample your data point across your chromatographic peak gives you the good representation of your peak area under the curve. Right? And you go back to the textbook, it means you need 6 or 8 to 10 points across your peak. Right? So that means if you have a 20 seconds wide peak using classical chromatographic system, means your cycle sampling, point-to-point sampling is 2 seconds. Right? If you're using fast LC faster, then you have to sample even faster. So the question here is keep your duty cycle and make the right trade-off in that you get the number of measurements on the MS scale that eventually fit or match your chromatographic scale. So PRM compared to SRM, right? We have the systematic measure of a targeted experiment. We have the limit of resolution. So what we want to do here, and this is summarizing what I said this morning, the high resolution gives us more selectivity, better discrimination of the analyte. We systematically use or collect MSMS spectra, which allows us to identify our peptides by mass of the precursor and the fragment very precisely. And as we see later on, we not only use the MSMS spectra in terms of M/Z dimension, we also use the intensity or relative intensities as a fingerprint to make sure we're measuring the right peptide. Trapping capabilities, I showed you that actually you can use it at your advantage and tune it or tweak it in a way. If you have a limited number of analytes, you can really get very good sensitivity. And you can multiplex, that means collect multiple precursor, fragment them collectively, and measure them. And that's a way to improve the number of peptides in one experiment. So I've shown it before, so nothing to add to this again, right? Again, just to repeat here, the spectra, you check first, and then you collect the traces to do the quantification. And obviously, as I'll discuss most of it in a quantitative way, most of the experiment we'll do here is with internal standard. And it is really true that if you want to quantify precisely using a stable isotopically labeled peptide in a calibrated way in a high degree of purity, it's quite expensive. And the numbers was $300 or euros this morning. It is somewhere in that figures. For some of the experiment where you stay more on a semi-quant way, or you just use an internal standard to calibrate across samples, the purity, as long it is 80 or 90%, give you a baseline. You cannot precisely, or you cannot accurately quantify, but you can precisely and reproducibly quantify across many samples, as long as you make sure you add always the same amount of internal standard. And I will stay with this mode today, and then eventually what it means if we quantify, we're looking at specific items for the internal standard as well as those for the endogenous peptide. So again, sorry to repeat myself, but now it is really the entire workflow. As we design the experiment, we design two methods. One, we could do them at the same time and just keep them separately, but one is uploaded on the instrument where you put the mass and the retention time window where you expect your peptide, you collect the data. We use a reference library here, and I'll come to much more details in that later on, to check first the identity, and then you use the second method, which is basically, as I explained earlier, you have an extraction window, you select the main ions, typically 6 to 8, you want to extract, and you process it with Skyline. So that just summarizes what I said. The important point here for all the experiment I'm describing is this module here is based on the reference library. And the reference library has, it's not an absolute necessity, but it makes life much, much easier if the spectra have been recorded on the same or similar platform under similar or identical conditions. Because then we have same fragmentation pattern, and we can really use the relative intensity, other fingerprint, and compare things, and have very stringent criteria. So that means for our studies here, we have the internal standard. So we have measured independently them and collected the spectra, sometimes at a different collision energy, and we keep the best one. It is very controlled in terms of gas pressure, collision energy, and if data have been collected in a preliminary discovery experiment on the same platform, we can use the MSMS spectra from the discovery experiment to drive the PRM experiment. We need to define elution times, because if we want to schedule, then it is quite important we control the chromatography and the peptides stay on schedule. And in most of the cases, as we have the internal standard with some good estimation of the purity, we also can put into account at least an estimate of the response factor. And as Tina has mentioned this morning, response factor could be quite different, so it's not necessarily directly proportional to the amount of sample, or the amount of peptide in your sample is not directly linked to the amount of signals, right? So you know, if you have a peptide with a poorly responder, it's not necessarily it is a low concentration, it just has a poor response factor. But knowing that upfront allows you, when you build your method, to adjust or have field times which are more appropriate for these kind of questions. So all that is basically done, it can be automated. It generates two methods, the first being uploaded on the instrument, the second one being used subsequently to process. And also again, on the reference library, I think I already said most of it, right? So we discussed that this morning. If we want to use a fingerprint and the relative intensity, we want to have well-defined parameters in terms of collision gas, which basically you have to make sure, have to test that you have the right setting and it is reproducible over time, and then you have a defined collision energy. So I've shown similar spectra or tracers this morning. That's the first benefits of doing PRM, right? You have better separation, you get signals, you can use them. So that, I think, it's pretty obvious. And the way you control your selectivity is either you use a higher resolution on the orbit trap, which means longer time, or you can play with the selection of the first quadrupole, which is in the latest version, second quadrupole. It allows you to go to 0.5 or even 0.3 AMU to get the selections. However, you have to be careful to your system is well tuned and calibrated M over Z, because that could cause loss of transmission of the precursor if you're not properly set. So, but it is doable. You can significantly decrease the selection window, increase therefore the signal to noise of your fragmentation subsequently, but you have to be careful to make sure your system gives you a good transmission. Just one more example, measuring in both modes, right? Even here, the results are similar. You'll see the consistency and the CVs in PRM are dramatically improved compared to a classical targeted experiment. And here, as I try to show you here, is that we can systematically quantify. So we systematically, in a targeted experiment, systematically target a precursor and some fragments, but having a signal doesn't necessarily mean we can properly quantify, right? because we have to deal with the interferences. We have to make sure we can see we are above limit of detection and above the limit of quantification. It's quite an important differences here. If you see a signal, the general rules is you'd call it detectable if the signal to noise is 3 to 1, but you should or you could not or you cannot really quantify it unless the signal's noise is 10 to 1, right? So detected doesn't necessarily mean quantifiable. And often, if we see at some of the previous example I showed here, right here we can even discuss if this is detectable, probably because the noise is such a way, but you need a reasonably signal to noise to be able to actually try to quantify or call it that way. So in SRM, this was an example of 10 peptides in plasma. So long story short, the plasma was depleted for the two most abundant protein, albumin, AGG, tryptic digested. And we had 93 peptides of interest, so they've been spiked as internal standard, and then we're measuring the internal standard, the corresponding peptide in SRM mode, and having quite stringent criteria to actually quantify it, signal to noise, and in principle know of, or at least the best we can say, there is not interferences. We call it quantifiable. Otherwise, we may just see something, but it would mean the signal is either very poor or the signal to noise is we may detect but not quantified. So if we apply the rules here in SRM, we had for this experiment, we had 10 control, 10 disease, plus everything duplicated, a success rate of 54%, and with an optimized internal standard triggered PRM, we can get to 84%. Right? And that's explaining how we do the experiment. But you know, if you look at that picture here, in the disease cases where the proteins are typically higher abundance, you see that we can actually much better quantify them, and we don't missing too many, right? There's just a few which are absent or too low we cannot detect, but most of them we can quantify. And here the success rate is much lower, but in that instance, that means the peptide in the control is very low, the endogenous peptide. But we know from the internal standard we should be able to detect it. So it's quite a dramatic improvement if you see at the coverage and the reliable and quantification result you can get comparing the two methods. So the design of a PRM experiment, I think I can go very quickly because it's in essence the same as an SRM experiment. First, the selection of the peptide, it has been discussed before, right? So it's all the rules of proteotypicity, having reasonable sequence, unique, good LC properties, good MS properties, and we are interested in quantification. So you want to make sure your peptides are stable, they are digesting well, there's no truncation, there's no Smith cleavage, just all these rules you've been discussing before. So all that applies, nothing new for PRM. You just take your same old biochemistry and bioinformatics you had, and you apply it. The acquisition method, I already mentioned that several times. The parameters are very few, right, for the specific to the peptide, and we need to add or adjust the parameters of the instrument, which might be more specific to the individual peptides, basically field time and resolution, as we have already discussed, and now I will show you how critical it is to get these parameters right. And in terms of data processing, that we have seen it before as well, right? First of all, you check that you have the right fragmentation pattern. At the same time, you could check in your spectra if you have foreign contributors, that will point or give you a hint that you may be more careful, and then you'll do the quantification by extracting the traces and doing as you do for an SRM using Skyline. So this is again the entire workflow. Method 1, you upload to the instrument, and this basically the table. You load all the instrument collision energy, you put a field time or max field time, which might be reached before. So this is basically the maximum which you try to couple with your orbit trap transient time, and the resolution of your MS2, and you have the options, or you can change, if you wish, the isolation window of the quadrupole and keep by default one m over z value. So that we've seen now earlier today, sensitivity, selectivity go hand by hand on an Orbitrap instrument, because if you fill longer, you get more ions, and at the same time, your transient is longer, so you can increase the resolution and the sensitivity at the same time. So you just have to balance between the scale, number of peptides you want to measure, and the degree of performance you would like to achieve, while keeping in mind you want to balance your signal in the chromatography dimension. So you need a cycle time, let's say 2 seconds, and then that determine basically how many events on the MS side you can have during that period of time. So the question is now balancing basically scale and performance. And here, typically, you want high performance, so we discussed earlier. You want to do a classical quantification experiment, so you really focus on precise quantification, best MS performance in terms of selectivity, sensitivity, and so on. And by definition, that would put a limit to the number of peptides you can measure, especially if you try to lock some of the parameters on your instrument. What I was alluding at the very beginning of this lecture is what we try to do is more what we call screening experiment, where here it is a focus of getting hundreds or even maybe 1,000 peptides. Oh, now it works. Sorry. 1,000 peptides and really leverage the multiplexing capabilities. Now, the question is, how do we balance these things? And a couple of examples to illustrate that is we have here a large-scale experiment, and I'll show you the data in more details during this talk. But what is done here is 600 peptide pairs, and 600 pairs means we're looking for the endogenous peptide and its corresponding internal standard. So that means the experiment means 1,200 peptides being measured. And I'll show you the data corresponding to that. And here is a smaller experiment where we did only 150. We restricted ourselves in terms of number of peptides, but we tried to get the best instrument parameters in terms of signal to noise and duty cycle. So here is a couple of examples to illustrate that. If we do this way, right, and we look at the cycle time, if we put more peptide and we keep this parameter fixed, 60,000 resolution, and that means 120 millisecond transient time. So to adjust the field time or be compatible, 110 milliseconds field time. So the things is synchronized. You see here, you see clearly the signal of your peptide. In some case, signals to noise is probably reasonable, but clearly in terms of quantification, it's pretty lousy because you get 2 or 3 points for quantification, right? So that's certainly okay to see. I can detect my peptides. I can believe they are there, but you can't properly quantify because you're not within the constraint of a quantification experiment. On the other hand here, with these parameters, with fewer peptide, we keep our duty cycle reasonably good. So we sample properly, we have good ion statistics. So this is the experiment which is successful, right? So you can properly quantify with these parameters, let's say it's 200-300 peptides, but after that you get in troubles. The second point is here, if we now look at another set of parameters, try to get here more peptides, we decrease the field time, and so means decrease the resolution. So now the cycle time is more appropriate, right? You quantify almost correctly, but, and that was a question we had over lunch, or somebody asked me about signal to noise and where is the limit. here you clearly see now you don't have a lot of ions in your ion trap, so you can no longer really do signal to noise or assess it, right? You can detect the larger transition, but here if you forget about really what it means because you are at the limit, you don't have enough ions anymore in your system. So what we did here, we decreased the field time and the resolution to cope with the cycle time of the chromatography, but then we lost on the mass spec side, right? So you clearly see this balance, right? You push on one hand, you lose on the other. So, and this is now just to show, it depends on the instrument, the numbers, not the absolute numbers, but the relation to the numbers, right? If you decrease the resolution, you have to adjust your field time, and then it tells you how many peptides you can have here. You could typically have 6 to 10 peptides in your experiment, and you will be reasonable cycle time. Now here you could put many more peptides, up to 60, but we will have a little bit of limitation on the signal to noise. Right, and that is what basically I already showed you, right? If these parameters, you're okay in terms of quantification. In this case here, probably I can call a peptide detected if the signal is reasonable, but certainly you cannot use it for quantification. Now the real question is, could we do these two things at the same time, right? It sounds like a difficult challenge, but that's what we try to strive at, is try to do performance and scale at the same time. So can we do that? And basically it means we have two experiment setup, a first one and a second one. One allows you to see many things, and the other one show you quality data, even the signal is not that high. I think I can pretty much skip that because we've discussed it many times. We need to match the constraint of the mass spec and the LC. We have said that. And the main challenge here is not so much to control the mass spec, because the mass spec does what it does. If you tell him 100 milliseconds, he will do 100 millisecond fill time. But a chromatograph or a separation or chromatographic is much more variable, right? If your peptide is eluting at 25.4 minutes, it doesn't need much in terms of either contamination on your sample or small changes in your chromatographic conditions that it shifts by a minute or half a minute, right? So getting the chromatographic right and reproducible is actually the challenge if you want to do scheduling, right? Because otherwise, if the variation is too big, you have to allow for larger windows, and that will really impact on the number of peptides you can include in one experiment. So that brings me to this really on-the-flight control or be more dynamic parameter setting. So there's a couple of levels here. So the first one here is exclusively related to optimize the acquisition time and control better the chromatography. And in order to do that, you take along the chromatogram a few peptides, which we call landmarks, which are peptides which will be used during the analysis to monitor that your chromatography is actually performing as it should do. That means if your peptide is supposed to elute, landmark number 3 is supposed to elute at 15 minutes, and all of a sudden it comes half a minute or a minute later, the system will notice and can correct for that. All the peptides scaled down the stream with adjusted with a delay because your chromatography has shifted. The second time is now if your chromatography is reproducible and you don't have to use the default in our hands, if our chromatography is reasonable, we would put 3 minutes, 2 to 3 minutes windows, and usually you don't miss too many. If you have the on-the-fly control and it adjusted dynamically, you can work with 1 minute or probably even less. So that allows you to double or triple the number of windows. That means the number of peptide you put in an experiment. And also, if we now look at the efficiency, if we look now for, let's say here we have a 2-minute window, the peak eluting for this period of time, that's actually what is collecting worth data. Everything else is just a system is scanning, collecting ions, and it's so-called wasted time, right? So the actual quantification time we'd like to use is this small window. So basically what we try to do here is, using on-the-fly correction, get much more efficient use of the instrument time and collect quality data when we have the sample eluting and not just looking left and right where the things could happen but may not happen. So we have already discussed that the first level, we try to monitor a few landmarks and we try to make sure the chromatography is on time. But what we try to do here is now really use the internal standard we're adding to the sample. And remember, I said from the beginning, we really try to do quantification even with not necessarily high purity peptides, but on a large scale using internal standard. So the idea here is that we have the internal standard in a defined amount, and we'll add it to the samples. So we know they are in the sample. We don't have to ask ourselves the question, are they there, or maybe they're not there, could we or can we not detect? We know the standard is there in a significant amount, so we can properly detect it. So basically what we'll do here is we'll monitor all the peptides of interest using the internal standard or proxy and using two distinct modes. One is a watch mode, so it's a quick scanning mode with a limited resolution just to detect. And once we detect, we switch to a so-called quantification mode when we can actually precisely quantify it. So if you remember the slide back here, if I can get it here, yes, right. Detection mode, we just want to see if something is there. If this is, we switch and we try to quantify properly. So we have this toggle between the two modes. So I already mentioned the problem of getting the chromatography under control. We actually developed it and we published it several times, first for the triple quad. And so this is all basically recycled in that mode here, this dynamic control of the elution times. But the really novelty here, which just came out a few months ago, we describe the so-called internal standard triggered PRM experiment. Right? And the idea here, we follow with low resolution, relatively short field times, the internal standard, and follow, I'll show you in a minute what it means. That means we measure the spectra, we compare to a fingerprint, and then, if we are confident, we switch to the so-called quantification mode, which is higher resolution, longer field time, and where you can properly quantify. So I don't want to go in too many details here. It is basically just showing the, the classical workflow. You have the method, it's loaded on the instrument, you measure, but what is added here is this dynamic control where as you measure, you have the MSMS spectra, you extract the data here, you compare to your reference library. So that's why the reference library is quite important, and I insisted on before. You on the fly could then adjust the parameters. That means switch between low, quote unquote, low and high resolution, right? Low resolution being at least 50,000, so it's not that low. And then the system keeps going from cycle to cycle. So this is just a cartoon to illustrate all of that. It is one peptide. This is in blue, the intensity, the reference spectra, right? So the peptide, which has been measured before under the same condition. And this is the elution window. So it is on schedule to come. So we monitor now every scan, every cycle, every 2 seconds. We look at the signal. So he has nothing to detect. Red is the actual peptide or precursor selected. So you see a first fragments, monotonous are appearing here. If you move, you see the signals are more and more intense. If you still do a spectral matching or comparison, it's still this is not perfect, right? It's far away. There's a few indicators, but it's not sufficient. But when you reach the point where your spectra are reasonably good agreement, and you put a threshold here, that means here, then you say, I can start, I know my peptide here, there's the right peptide, I will start to quantify it. And you switch to the so-called quantification mode. So at this point, the system shifts to the other parameters, and it also selects the precursor of the endogenous peptide at the same time. Now, I will collect high-quality data across this peak, same for this one here, and I can properly or use that data for subsequently properly quantify. And make long story short, is basically, I don't want to go in all the technical details, but that cartoon I showed you before, right? You're pretty inefficient here in terms of data collection. If so, you calculate the actual measuring time where you collect reliable, solid data. It's a 10-20% yield. If we do it now, we see we have a watch mode which costs very little time because that's go very fast. And when we switch, then we collect the data. So if we actually collect and calculate it back, you know, the collection efficiency, time spent on measuring the real stuff, is close to 80 or 90%. I try to illustrate that with a couple of examples, and all these examples are in the paper I just mentioned. And I'm not going into the all small technical details. You can read it in the paper if you will, but in essence, the experiment, this is actually the 93 peptides, pairs of peptides I showed you before in this blue chessboard, blue and gray, right, which are corresponding to 50 proteins. And we actually did a comparative study where we look at the regular PRM, so that just default parameters as described earlier today. The improved method where uses the dual switching mode, and we also have the data collected in SRM. So as I said, plasma, a simple depletion, 10 and 10, and we measure here the triggering by the internal standard. And these are the PRM for the regular standard PRM experiment. We're using both same parameters here for the SPRM, for the watch and the quantification mode, right? So the quantification mode, which is also applied for the, and the watch mode of the internal standard is what we would use here. So I'll come to the processing later on. So this is now just one, actually two pieces of information on that slide here. So first of all, on the bottom here, for one specific peptide, you have the data in SRM, probably suboptimal. In PRM, we see that we get the signal. It's pretty obvious that the ion ratio and some of the statistic doesn't really, ion counts doesn't really work out, but at least we can claim it is detected. Now, if we optimize to a quantification mode, you have a clear, nice signal, right? So, and I think that's pretty convincing in itself. Now, the experiment was the following. We had these 93 peptides. They've been spiked now at different concentration in the plasma, right? And this is the different amount spiked from the different peptides, and how many peptides you detect. I mean, looking at the different transition, typically looking at 6 transitions for each peptide. You know, if you have relatively high concentration, you get most of them in ISPRI. And PRM, as long as you have a lot of signals, not that bad, and even SRM can do a pretty good job. Right? But as soon as you dilute by a factor 2, 4 or more, you see that it's a drop with a classical method. ISPRM is also dropping because you also reach the limit of detection quantification. But it is at least to the 10, 20 attomole range, you still have a pretty good success rate. And I try to illustrate that with one example and walk you through that example. So this is one peptide, protein is PJK1, and actually two peptides reflecting that protein. So this is SRM, 6 most intent transition, PRM, 6 most intent transition, right? No further fine-tuning or processing, we just put it in Skyline, integrated, and here are the data. So, you have the control, the disease here. You probably see some trends here, but it's without question that the data is quite noisy here. It does quite a bit more convincing on the PRM side. If you take back the SRM and now not use the 6 transition, this hasn't changed, we kept the 6 transition in SRM, you see that does a much better job, right? You get a more consistent picture. Nonetheless, you still have some significant questions that at least put you a few red flags. This is the slide I showed you exactly before. I just now put it on the full screen. And now I'm going to look at another peptide of that same protein. So we're adding one more peptide, and it doesn't fit that well, right? So this exactly. So there's a regular PRM experiment. If I look at the gray, the ion ratio, not the ratio, the factor response or the response of the different peptides are not coherent, right? You see really large differences and inconsistencies. So that, again, put a red flag here. And why is that? It's even it is PRM, maybe we're not optimum into our acquisition. And just as an example, look at this measurement here. You know, this peptide is not maybe not as responding as well as the other one. You see the signal is clearly there, but in terms of signal to noise or ion statistic, it is questionable, right? You clearly detect it, but you can't use such a signal for quantification. So even putting a number here is probably at the edge what it could do. You could see detected, but trying to quantify is questionable. Now, this is the same sample, remeasured now using ISPRM. And if you look at that same peptide, now you have a proper quantification, right? Because you have longer time, your cycle time has been adjusted only during that period of time, and you can now trust your ion ratio. And that is not only true for that peptide. Across the entire data set, you see that you have a really good and coherent data set. And now this is a summary slide. So this I've shown you before, and it is CSRM, the classical PRM, 62% versus the internal standard PRM with 84%. So you see the increase in data quality if you add this additional step into the system. So to summarize that modules here is that we have a dynamic setup which basically does on-the-fly decision and corrections. It has two levels. First of all, it checks that the chromatography is set properly and it is reproducible. And if there are drift within some tolerance, everything is 5 minutes or 10 minutes off, right? It will stop the experiment because something is really wrong with the HPLC. But it is within the boundaries of your chromatography. It would correct and adjust on the flight. And then in terms of measuring the peptide of interest in the targeted experiment, what you do here, you use two different modes of acquisition. You first follow your internal standard. When it is there, you assure the right it's the right analyte, you switch and you quantify, right? And that's why I insisted early on this morning that creating a database of quality is important because you use it as a guide for doing your experiment and doing the flight. So the second experiment I'd like to briefly discuss here is the experiment where we have tried to scale up. And why 606? That basically we had a number of projects running in urine, plasma, and others, and some synthetic peptide we had for other purposes. We basically cleaned the fridge and we found 606 peptides. We can dilute, aliquot, and then use as reference. So we spike them in different samples. So as the samples are heterogeneous, you not necessarily expect to see the 600 peptides in any sample because they're different origin, but it means you can trigger 606 peptides, and this is a different sample we measured, and we did the comparative study in SRM, PRM, and again, these parameters described here as very similar to those which I showed on the previous slide. And actually the slide I showed you early on, right, the 600 pairs here. So now this is done where we have now 60 milliseconds, 110 milliseconds field time, 3000 resolution, 110 milliseconds field time, and we can properly sample 600 peptides in a way very similar where we had before in a regular PRM way only 150, right? So the idea was, can we get this quality? Remember the question, marry quality and quantity. So now we can get quality on a larger scale, at least 600 peptides, so factor 4 And probably if we tweak and optimize it further, there is still room to get a few more. So I'm not saying we can go to many, many thousands, but probably double that number is probably within reach. My system is not answering. So this is one more example to illustrate all of that, right? So this is the number of peptide detected in plasma. Remember, we did the experiment in plasma, in urine, and HeLa cell. So we're not expecting to see 600, but about half of them should be present, or nearly half of them. So we detected and quantified using ISPRM 250. That's what we could get with the setup we had. But if we look at classical method, either geared to scale or to quality, you get at best half of them, right? And then you can even replicate the experiment several times and measure CV. I have to recall if it was a fourth or fifth replicate, and then you measure the CV. But here, in any case, ISPRM, you get a very good reproducibility of quantifications, a CV below 10%. In that case here, you see the peak shape, and the sampling rate is not perfect. So the variability is somewhere around 30% average on all these peptides. And in the method where we could just detect, I mean, there's no way to even try to quantify and give a CV, because this is, at best, we can call it detected, but we cannot quantify. And I think that becomes an important question here. That was more or less a question I had this morning. What about limit of detection and signal to noise, right? We can detect a lot of things, but we cannot necessarily quantify everything, right? And we have to be careful, really looking at the raw data, or at least at the traces, to make sure you are in a scheme where you are allowed to quantify and not you're just doing a wild guess based on some few points you may have seen. So this slide summarizes the entire experiment. So you have the different samples in which the peptide have been spiked in. This is one example what you could see. So it could be elevated in one sample, missing another one. This is also a type of data shows you that you could get very strong signals if it's very abundant in one sample. Here, this is all relative numbers, and basically the signal happy to get the same box times 300 times 5,000 here. But you see, even it's fairly low, you could still get enough counts, so you get somehow reliable measurements, and you can actually use the data and you take a quantitative measurement out of such a data set. And it shows you the kind of dynamic range you can expect in this kind of experiment. So I, more or less, coming to the end. I think I have shown you that slide a couple of times. It kind of summarizes the entire workflow, and it is in some way a change of how to do a targeted experiment, in that we have a method for the acquisition, which is actually straightforward from what is needed in terms of intrinsic information related to the peptide. Then it's uploaded on the system, which has a relatively elaborated way at looking at the data as they are collected to actually drive or set the experiment to do more or maximize the output. And it is all based on the fact that we have here full MSMS spectra. We have a reference library with the fragmentation pattern of reference, and we systematically identify first the analyte, may even see if they are contaminant, and then use that information to actually dynamically go in the quantification. So it's not only a change in that you quantify and then you validate your measurements, like in SRM by looking at ion ratios. Here it is a switch in that you first identify, and once you know what you measure, you try to quantify. And the second shift here, you don't do it post-acquisition or in the course of the process, you do it live on the flight, right? You know your peptide you're expecting. If it comes, you check that it is there, and then you collect the trace and you quantify it, right? And the quantification is then eventually done. You extract from your MSMS spectra the traces post-acquisition, and then you apply exactly the same software tools you used to do for an SRM experiment. Basically, what we have been working with Brenda and his group is actually write a script that the data are converted to what we call an SRM-like format. It's basically a small SQL database or file, and then it is will be compatible with Skyline and will be processed exactly in the same way, pretty transparently. And that is what I just said, summarized here. You have your different experiments, you have the raw files, the script at the end of the measurement extract typically 6 to 8 ions, but there is no limit there. You put the master layers, as we discussed this morning, because that's an important factor. The good news is here, if you set something suboptimal, you don't have the right tolerance or the right ions, you can always go back and reprocess it, right? So it is iterative process. You don't have to measure all the data on file here. We convert all these traces in SQL, and the reference library is also then be used in Skyline for the subsequent validation. But basically, this file or this sum file together is sent, and this is one example for one data set where you basically see output one peptide across many samples. That's the typical kind of output you will get, and you can then use it to QC and eventually collate all your data together. So I will try to summarize on this point here, and I hope I could, with a few examples, show that we have a method here which can overcome some of the typical limitation looking at complex biological samples or clinical samples. That means high resolution give you an additional level of selectivity and sensitivity using the trapping device. The technology, when we're talking PRM 3-4 years ago, there was kind of an exotic topic. I think it's now slowly getting more traction. So I think it's still maturing. We need to understand better, and we slowly understand all the parameters and how to design an experiment. But it's clearly a technology that will probably get more accepted in the future. The way of doing experiment, the parameter setting has started to be streamlined. We understand how it works and which parameters are critical. At the beginning, it was, remember, the Orbitrap was a qualitative instrument, right? And the first Q-exactive were benchtop Orbitraps instruments that they were firmly thought to do DDA experiments. It's only in the last few years or 5 years that we slowly could implement them more and more to not only doing quantitative experiment, but quantitative in terms of relative quantification, such as TMT or SciLag experiment, but now in really true quantitative experiment where you have your internal stuff well calibrated and you can measure with very low CV. So I think we discuss the benefits in terms of selectivity, sensitivity, the accurate mass. We really know what we measure. We're not guessing too much anymore. The method is fairly straightforward, and the data processing is actually also fairly straightforward, right? It's if we have the script implemented. They take the data, even though there is some complexity in the acquisition type or different scan type, all that can be overcome. Then we can easily apply the method and use it systematically. And it can be used in clinical samples. We showed now a number of examples. We can do that in biological very generally and quite successfully. And the fact that we have internal standard, there is a cost associated to it, but also, as I mentioned, you don't necessarily need the high-cost internal standard to do such experiment, and those peptides are coming more and more commodity. You actually use them to drive your experiment, and you're not doing post-acquisition. So I have to thank all the people in the lab who did most of the experiment, especially Sébastien Gallien, who did all the experiment on the ISPRM, the clinician who gave us some of the samples. We also closely collaborating with people at Thermo Fisher, because as you see, we have to interact quite a bit in terms of software, having access to some of the code to be able to prototype by using some of this program. And last but not least, I'd like to thank the funding agency, especially the Fonds National de la Recherche, who sponsors my lab, and also the Ministry of Research of Luxembourg. And I'd like to thank you all for your kind attention.
So fantastic. Thanks a lot, Bruno. Are there some questions? Yep.
Thank you. First of all, you have mentioned that you have at the last you mentioned that you were collaborating with Thermo Scientific for software. I mean, you have write a script to integrate it in the QExactive.
So what we have done, we had access to part of the API, so we could prototype the codes. And even in the paper we published, we release the general principles. There is an executable which allow to in principle replicate that. What is going to happen now, this is going to be implemented as a standard piece of software in the next release.
Okay, it was my question.
The next release, I'm not Thermo, so I can't give you a date on that. You have to ask to your sales rep.
And the second question goes, follow the first one. Which criteria do you select to decide switching between low resolution and high resolutions? Because when presenting your curve, the starting of the peak.
Yeah, so that's a very good question, and if I can go back. No, I can't. I lost the signal, so I don't know why. God. Are you coming back? Okay, good. Maybe there's a delay, so this is much slower. We can get back. Here. Yeah. So this is a slide. So the key of all is the reference library and the reference fragmentation pattern or reference spectra. So this is what you expect if your peptide would. And you see as we start eluting, the concentration and the larger peak coming, right? And here you have more ions. But so the two things here, you need to get a good match in order to say my peptide is the one I want. Now to have a good match, that means two spectra have to be similar or very similar, and you can, the mathematical ways, there are different ways, dot products, or look at the angle. So there is different mathematics you can apply, they're more or less equivalent. But to be equal or similar, they have to be acquired under the same condition. And that's why I was insisting at the very beginning, you need your spectra to measure it under the same condition, collision energy and similar pressure, so you have, or you could reproduce your spectrum, right? Otherwise, if it's only based on presence, absence, you have a much more difficult work to do a call, is it the right peptide or not? Right? Or you have to have a different heuristic, say, when I see 3 out of the 4 ions, and they more or less are in the range I go, right? So here we're quite stringent in that, but we also know, we also have other benefits. We don't have a lot of false triggering, right? You could relax your rules, or you could have a lower quality reference, but then you would have a lot of more of false positive events, right?
Thank you.
Yeah, so I would follow on the false positive questions. So now that you're going to thousands of assays measured per runs, do you still visually check everything in hundreds of runs, or do you have any way to estimate the false discovery rate of this detection?
No, so this is for the acquisition, right? Once you have collected your data, you have all the MSMS spectra, so you could systematically, either manually or automatically, write a script, because now we detect the spectra. Actually, it's a suboptimum detection of the spectra, right? Matching here. If you match there, it's already good. It must probably match up there much better, right? But this is the internal standard, which is known to be there. The endogenous peptide, which would be lower or more noisy here, even if it's a match of the internal standard, doesn't mean it's a match on the endogenous peptide, right? And you could detect or quantify that one, doesn't necessarily may detect the endogenous, but you may not quantify it, right? So we don't necessarily go with false discovery rate in that sense, but for every measurement, we would put criteria. Is the MSMS spectra of the endogenous peptide within a tolerance, and then is still your coelution of the your classical SRM QC still apply, right?
Just to make sure that I understood it correctly, your reference library is created based on stable isotope labeled peptides, or?
That's the way we did it in that experiment. If you would have done a discovery experiment on the same platform, you could use your reference peptide from the discovery experiment. But then you have to, actually we did that experiment, we're writing out the paper, you have to put an extra level. You cannot just dump mascot search with that score up. You have to make sure, and also to see that you don't have too much coelution, and so we put some more additional annotation. People peak have to be annotated, and if you have too many extra generous peak, you don't want them, because then again you may detect or trigger in the wrong way, right? But it's not it's not an absolute necessity to have the isotopic label. But then, if you use the endos, then you back in the problems. You detect the endos only if it is there, and this low concentration is not, right? The point of having the internal standard, you spike it at a level which is close to where you were quantified. That's what you said this morning, right? But you make sure it is at least in the level you can properly, it's not at the edge, right? So if we quantify the edge, we would still spike it at least a log above to make sure it doesn't get slip through.
Okay, further questions? Okay, so if not, I think we're going directly into SkyJam now session, right? So let's meet back upstairs in 5 minutes, and we'll start the SkyJam tutorial session. So let's thank Bruno again for these nice talks.
Thank you.